All right, thanks for coming to my talk today. I'll be very excited to share a recent project we did on characterizing tree space of Bayesian phylogenetic inference on large viral data set. So phylogenetic analysis has been used extensively to address important questions, including evolution relationship of SARS-CoV-2 variants, origin time of SARS-CoV-2 clades, and time-dependent effective population size. It can also be used to map key character evolution events over the tree, including molecular and phenotypic traits, as well as the geographic dispersal history. Currently, the most popular phylogenetic inference approach is the sequential approach, using tool-specific design for each step. So in the first step, we infer an unrooted phylogram based just on the sequence alignment. Then we incorporate additional sampling time information to convert the unrooted tree into a rooted time tree. Finally, conditioning on that time tree, we can estimate other downstream parameters. Alternatively, another approach is to estimate everything jointly in a single framework, Bayesian framework, using all available data. This joint approach expresses and accommodates uncertainties naturally and provides a suite of tools for model assessment and analysis diagnostics, and also allow us to incorporate various sources of information through PAR. However, all these nice properties are just immediately outweighed by this one simple factor. This joint approach is just impractical for any data set with more than a few thousand of sequences. But why is this Bayesian approach so slow, right? So recall that in Bayesian inference, our focus is the posterior, computed as the product of the likelihood and the prior. And the posterior space is typically approximated by the MCMC, the Markov chain Monte Carlo, where we usually run some number of replicate chains with different starting points. And then compare the sample to collect it to ensure the convergence of the MCMC algorithm. So understandably, Bayesian inference should be slower, right, than the maximum likelihood, as we're trying to infer the entire distribution instead of just getting to the highest point. But when the posterior space becomes more complex, for example, being multimodal, or to an extreme being super rigid, where there could be multiple plateaus separated by valleys, terraces, and ridges, inferring the posterior distribution could be increasingly more difficult or becoming almost impossible. So in this study, we first want to ask, what does the posterior tree space of Bayesian phylogenetic inference actually look like? So to address that, we compiled 15 classical viral data sets that are relatively large, representing various different types of viruses. And these data sets were also selected to cover various tree shapes. Here I'm showing the MCC tree of each data set in each panel. As we can see, these trees usually consist of some rare early samples, and many recent samples collected rather close to the present. So for some of these data sets, when the MRCA is much older than the tips, this looks somewhat similar with a conventional species-level ultrametric tree. But most other trees actually look quite imbalanced, especially for the human flu trees, presumably due to high lineage turnover. One other major feature of this data set is that most of the branches here are only weakly supported, which I guess not really surprising given the limited amount of information in the sequence alignment. So as shown in this figure, you can see most branches in the tree, most sites in the alignment basically entail no mutation at all when you map them on the tree. And this makes the viral sequence data set quite distinct from the species-level coding sequence data set which most phylogenetic methods were originally developed for. All right, so our expectation is that the posterior are going to be quite diffuse and potentially difficult to sample. So we ran 10 independent Bayes MCMC replicates for each data set, including 5 long runs with 400 million generations each, to kind of imitate the best practice in reality, and all 5 even longer runs with 1 billion generations each. So then let's look at the result. First, let's focus on the convergence of tree topologies among replicate chains. So to realize the tree space, we first compute the pairwise tree distance matrix, and then use multi-dimensional scaling to transform that high-dimensional tree space onto a two-dimensional graph referred to as an MDS plot, in which each dot represents a tree. And then here's the posterior tree space of each data set. Each dot represents a sample tree, while its color indicates which MCMC chain it's sampled from. So a clear separation of the colors indicates lack of convergence among chains. That is, for many of these data sets, each individual MCMC comes stuck as some local modes are not really able to commute, even after 1 billion generations. And even for the ones who roughly converge, the sampler tend to experience difficulty in mixing within the chain. So it appears that the posterior tree space is really quite complex, with most of them look multimodal and many contain terraces and ridges. So most of the computation time in Bayesian phylogenetic analysis actually is spent in exploring, or rather say wasted, in wandering around this complex tree space. So to make this observation more clear, let's take a closer look at some example data sets. Recall that each dot here represents a sample tree, and below I'll show an animation where dots appear sequentially to reveal how the MCMC sampler progresses in the tree space. So here is a result of an Ebola data set. Each panel here corresponds to an MCMC chain. I have to warn you, this looks perfectly smooth on my screen, but not really working here. So here I'm showing actually each dot appears sequentially as indicate different sample trees. And the top row corresponds to the 400 million generation chains, while the bottom row corresponds to the 5 longer billion generation chains. And the rainbow color of each dot is just used to indicate the chain progression. Appears that some chains, especially the shorter ones, fail to reach some of the modes, and occasionally a chain commutes between the modes after a very large number of generations. Now let's put all the replicate together. We can see the posterior tree space kind of mimic this cartoon distribution in that the sampler only crosses the valley rather infrequently, possibly through some narrow bridges. And here is another example, the same type of animation, but for the Lhasa virus data set. Again, it should be much smoother than that. And so we can see there appears to be 2 modes. And basically, each replicate chain is stuck on one of them. Very rarely, some chain were able to move to the other mode, but then just stuck there again. So is the convergence ratio really that bad? So let's look closer at the inference of this data set. It turns out that these failures are all caused by just 2 taxa, shown as the purple and blue tips in this part of the MCC tree, out of all the 551 taxa. You may have noticed that the posterior support of most nodes in this tree, in this clade, are just about 50%. It turns out that corresponds exactly to the 2 modes identified in the left MDS plot. And here, the focal sequence is the purple one. So basically, in half the range, it's inferred to be sister with the orange clade, while in the other half, it's inferred to be sister with the blue tip. And it appears that this is likely caused by a putative recombination event involving these 3 tips. As we can see here, basically, the first half of the purple sequence looks very similar with the blue one, whereas the second half looks almost identical with the orange one. Then if we remove the purple and blue tips from each sample posterior tree, suddenly the convergence issue disappears. And we don't see the separation of modes in the MDS plot anymore. We can also see the contrast before and after pruning quantitatively using diagnostic statistics. Here we compute PSRF, the potential scale reduction factor for both tree topology and parsimony score, and intending to quantify the between MCMC chain convergence behavior. And the PSRF values are indeed much greater than the acceptable cutoff before pruning. However, once removing those 2 taxa out of the 500 taxas, the PSRF become very close to 1, indicating almost perfect convergence. And so while now you may be wondering how can we identify those 2 taxa, right? So we could actually try to assess the MCMC performance of each clade and look at those clade-specific diagnostics. Here in the scatter plot, each dot represents a clade, and the y-axis indicate the between-chain convergence behavior quantified by the standard deviation of clade frequency, while the x-axis indicate the within-chain mixing quantified by the effective population size of the clade occurrence, the clade ESS. So the y-axis values above 0.1 indicate lack of convergence, while the x-axis value below 200 indicate poor mixing. Does the nodes in the bottom right quadrant have good MCMC performance? So let's hide them for now and focus on the nodes that perform poorly. As we can see here, only about 3% of nodes actually failed. And most of these failed nodes are associated with this small clade. Once we prune the purple and blue taxa from the tree, all the nodes actually with convergence issues suddenly disappear. Then the recombination event can also be identified with site-specific MCMC diagnostics. So here we're basically treating the phylogenetic likelihood at each site as a continuous variable and then compute the corresponding PSRF and ESS as 0. And each dot here in this bottom scatter plot does represent a site. Y and x-axis indicate the between-chain convergence and within-chain mixing of each site respectively. Again, let's hide the good performance sites and focus on the 2% site who failed. When we list all the nucleotides of those sites for these 3 taxa, actually this recombination pattern sent out immediately. And after removing these 2 taxa, the sites also that exhibit convergence failure also disappear. So similar pattern actually observed for most of this data set in that a very small fraction of the clades and sites failed in terms of their MCMC performance. as shown here in this figure, highlighted by the bottom orange box. All right, so that's the first major factor we identified. Here I'm going to switch the gear a little bit. Going to talk about the other factor that may further exacerbate the tree inference difficulty for phylogenetic inference, which is the weak identifiability among trees caused by the interaction between phylogenetic and coalescent likelihoods. So recall here, this is our Bayesian formula for computing the joint posterior density. To develop some intuition, let's first look at a simple example. Here we have two trees, and data is just a single nucleotide. If we only consider the phylogenetic likelihood, clearly tree 1 will be favored, right? 1 versus 2 mutations required. But when all the samples are contemporaneous, the coalescent likelihood is uniform across all labeled histories, so which tree is favored is entirely determined by the sequence data. However, when the tips are serially sampled, the coalescent likelihood will not be uniform anymore. Instead, it will be higher for more imbalanced trees. So tree 2 now actually has a higher coalescent likelihood. So which tree will be favored a posteriori actually depends on the interplay between these two likelihoods. And let's denote the product of these two likelihoods as phi and realize difference in the log phi between these two trees as a function of the clock rate and effective population size. We can see that there is a certain part of the parameter space that the tree 2 actually is favored and vice versa. Well, that's just a toy example, right? So does this actually manifest empirically? It turns out it does in a quite striking way. So here I'm showing results of an H1N1 data set. Each dot in the scatter plot corresponds to an MCMC sample, whose x-axis indicate the relative coalescent likelihood and y-axis indicate the relative phylogenetic likelihood. And here the two likelihoods are actually strongly negatively correlated, demonstrating the interaction between them. And in addition, this also indicates that theoretically, we just should not expect the sequential and the joint approaches to produce the same tree topology. Indeed, if we actually add the result inferred under the sequential approach as the orange triangle here, we can clearly see, just as what you would expect, the maximum likelihood tree has a much higher phylogenetic likelihood, while much lower coalescent likelihood compared to the posterior trees. And this interaction pattern is widespread across all data sets we examined, and most of them exhibit negative correlation between the two likelihoods, as shown here. In addition, this interaction also affects biologically relevant parameters. Right, so this toy example also predicts that most imbalanced tree should be associated, the more imbalanced tree should be associated with a higher coalescent likelihood and lower phylogenetic likelihood, and then tends to estimate higher clock rate. So we would expect that clock rate and imbalanceness to be positively correlated with coalescent likelihood, but negatively correlated with phylogenetic likelihood, which is exactly what we observe across all the empirical data sets. All right, so does the convergence issue actually matter for the focal parameter we care about in phylogenetic analysis? Well, it appears sometimes they do. So here we look at the number of dispersal events inferred between two focal geographic areas in this important study investigating the early spread of HIV. So for some of the MCMC replicates, only a couple of events is inferred, while for some other replicates, the estimate is at least 6 or 7, could be even higher. Right, so this observed impact is kind of to be expected, given that the character mapping history is likely to be affected by local topological structure. However, to end on just a more positive note, the impact on effective population side is basically quite negligible. As you can see here, demographic trajectory inferred across MCMC replicates are barely distinguishable. All right, so just here the take-home message, but I don't think I have time to repeat. I would like to definitely briefly mention something I don't have time to cover here already, which is the typical data quality tests like root-to-tip regression generally do not have that much for resolving the convergence issue. So to really address that, we may actually need to design new diagnostic tools, new MCMC proposals, and possibly a new maximum likelihood method that's actually joint inference. In addition, output from the very long Bayesian analysis could actually be used for training new machine learning methods in the future. All right, with that, I would like to thank Eric, other co-authors, Mike May for discussion, and my funding sources, and you all for listening. And I'm more than happy to take any questions. Thank you.
So thanks everyone for coming. I guess it's time. Yeah. Okay, today I'm going to talk a little bit about RefBase and new features, new applications that we have implemented in RefBase over the last couple of years. But before I start, I'll give a little bit background and a history. So Bayesian phylogenetics really started off in 1996, really seminal papers that showed how to do MCMC analysis for phylogenetics. And then pretty soon afterwards, there was Mr. Bayes, so it only took 5 years, and Mr. Bayes went from version 1 to version 3 in only 2 years. You will see that there's a time scaling effect now happening for RefBase, because already in 2004, there was the kickoff meeting for Mr. Bayes 4, that then led into a prototype in 2008. But that prototype wasn't really what was thought about for doing RefBase. So that was a complete restart in 2009 and redesigning it in what we now have as RefBase. So then there were several rewrites, at least 3 major rewrites where we started from scratch again. And in 2014, we actually had the first working version of RefBase. We had the first larger workshop where we exposed users to RefBase. Then in 2016, we published RefBase version 1. That's the official publication that there is right now. Then there were a lot of improvements, a lot of additional features. In 2020, we decided to call it RefBase version 1.1. In 2022, we went to RefBase version 2.2, and there's more improvements, more features, a lot of more things happening. At the beginning, this was the RefBase team that we started out with. These 8 people really contributed in different ways and really made RefBase 1.1 happening. But then since then, this is the new amount of developers, so there's a lot of people, and there's probably more than I put in on this slide, who actually have contributed to RefBase. And because of all these new features and all these new collaborators, we thought now it's almost 10 years later that it's about time to actually decide to release a new version, RefBase 2.0. It's not actually something that is a complete rewrite or completely different. It's really an incremental update, but it's also really to give credit to all the people who have worked on RefBase. I also want to highlight specifically 2 people. That's Joel and Ben, who have spent so much time on making RefBase much more usable, but haven't really gotten much credit for it. So far, it's really a lot of that time, until you get actually a release note. So what is RefBase and how does it work? RefBase is really built around this concept of probabilistic graphical models, which you can see on the left-hand side there. The graphical models was the idea that we can compartmentalize the model that you want to specify to run your phylogenetic analysis. And that is really the central idea, that we actually build it in computer memory. On the right-hand side, you see a short example on the ref script that you need to write to actually use that model when you run your analysis. So we have its own interactive programming language ref. So the strengths of RefBase are that it's extremely flexible because of this probabilistic graphical model framework. You can exchange small parts, you can replace them, you can extend it. That makes it very flexible, extremely powerful. It also is very explicit, so you can see in each part of the model what parameters there actually are, what are the prior distributions, what are the connections, so that is really pedagogical in that way as well. And over the years, it has tremendous functionality. There are so many different models that you can actually implement. I can't even list all of them, and there's probably half of the features are not even documented. So just ask people if you want to know more how to do these things. Then there's of course weaknesses, and over the years, since now more than 10 years we have been using it, our experience of the weaknesses are that it's very complex. If you see these things, these graphical models, if you see the scripting languages, if you actually see the inference script, that is just a small excerpt because it doesn't show what the MCMC operators are, it doesn't see how the MCMC works, so it's usually 3 or 4 times as long, and that's just the simple model. So it's kind of complex, and it scares off a lot of people in the beginning. So it requires more time to learn, and it requires more time to actually understanding it, because you just can't click a button, let it run. You need to know what you're doing. So the punchline is, if you have complex models and if you want to actually explore models, then RefBase is a really great tool that you can use. But otherwise, if you want to run a standard phylogenetic analysis that is really well established, maybe there's other tools that are as good, but you can still use RefBase, that's fine. Okay, so now I want to talk to you a little bit about what RefBase or what the range of analysis are, what RefBase can actually do. I tried to give that as a brief overview. So we start really from the recent time. We have been working a lot of these parts, that's recent time demographic and phylogeographic inference. In the middle, there's all the macroevolutionary and phylogenetic inference. And on the left-hand side, there's really deep time, a lot of different models for deep time analysis, tip dating, fossil studies. Okay, let's start with the demographic inference. You can use RefBase to infer the demographic histories. We recently implemented a stairway plot approach, so you can use your millions of SNPs, whole genome data for however many individuals you have, 50, 100, and get these really nice demographic histories. That is super fast. Simplest models run in 2 minutes on my laptop. Or you can do sequence-based approaches. So people are usually familiar with the Bayesian skyline plot. You have the sequences, you estimate the coalescence tree. From the coalescence tree, you get the demographic, your population size history, and that can use any present-day samples, also ancient samples, this kind of data. So zooming out a little bit from the single populations, we can go to multiple populations. We can do phylogeographic inference. Here's the example of what we in the lab actually have been working on, the fireflies. This was an office mate of mine working on grasshoppers, where we have 6 populations for the fireflies. We also have whole genome data. We use million SNPs and infer the timing and the splitting of these populations based on these polymorphism-aware phylogenetic models that Rui Borges has been working on quite a lot. They're super nice, and there's more work in that direction to come. So then zooming out a little bit more, this is an overview figure of what the phylogenetic inference can do. So you can use in RefBase any different type of data. You can use molecular data as DNA, amino acids. You have discrete morphological data. There were several talks about that already. You can use continuous data. There's a lot of different development on the character models because RefBase is so flexible. You can really design that for your specific data set. A lot of among character rate variation. We also had a few talks yesterday about that. Data partitioning is not that much talked about lately, but again, you have a lot of flexibility either by the genes or you do that by anatomical reasons, whatever you want to do. There's no limitations that you actually have in RefBase to specify these type of models. There's a lot of different types of clock models, relaxed clock models, and then you can include any tree priors. So this figure really goes about the tip dating for the phylogenetic inference. That's just an example of people who really have pushed that work forward. But the important message here is that because for tip dating and phylogenetic analysis, they are really complex, and there's a lot of complex models. You divide the data in a lot of complex different ways. This is where the strength of RefBase comes in, because you can fine-tune it and modify it exactly for these type of questions that you want to ask, instead of just chucking it into a predefined default analysis. Okay, then there has been a lot of the work of once we actually have the phylogeny or jointly doing it. So Michael Landis has been working a lot on biogeographic modeling. We had before the coffee break a talk in that similar direction, where you can use biogeographic models for infer the mode of biogeographic evolution, but also ancestral state estimation and use that in dating. We have trait evolution. Interestingly, there's a lot of discrete trait evolution models in RefBase with the tutorials, but no one has bothered to publish about them because they just fell out of, from the teaching. We also have a lot of continuous trait evolution. Priscilla gave a really nice talk about that yesterday. So from the whole range of Brownian motion to Ornstein-Uhlenbeck models that you can use. You can always use, the nice part there about RefBase is you can always use them for assuming the tree and learning about these models and their trait evolution, but you can also use them then in a combined analysis like Jay was indicating before, if you want to do joint inference to infer your tree with all the steps together. What I've been working on a lot is also on estimating diversification rates. So you can have time-varying diversification rates. You can try to connect them with some environmental variables. On the left-hand side, you see you have diversification rates that can vary among lineages, and you can try to infer the correlation with discrete states or also with additional hidden rate variation. They are a little bit slow, but those are complex models. We have worked a lot on model testing, so because RefBase is Bayesian inference, so most of the model testing is estimating marginal likelihoods and base factors, but we also have been working a lot on model adequacy testing, so Jeremy Brown and his group have really worked a lot on that. But we recently also have integrated into a leave one out cross-validations, which is much faster. You don't need to do anything extra, just run once your MCMC analysis, and you get the cross-validation prediction score. And then finally, we have Ref Gadgets. It's our R package. It's really nice. So all the output from RefBase, what do you do with that? Carrie, she worked on this R package for post-processing and for visualization. So all these nice plots that I showed you with character mapping and the trees, they were produced in that R package. With that, I want to thank it's a lot of different funding from a lot of different groups. That's just the major funding agencies that went behind RefBase. And all the people who have more contributed to it than probably twice as many other people. And thank you.
Welcome, everybody. My name is Lénard Szantho, and I brought you today a statement, namely compositionally constrained sites drive long branch attraction. We published an article just 1 year ago about this, and hopefully with my co-authors, who you see there, and hopefully by the end of the talk, you will be convinced that this statement is true, and you will use our method or other more advanced ones. So in the last 2 decades, after the drop in the price for the sequencing, today we have tremendous amount of sequences, and also computers got faster and faster, following the Moore's law. So if you compare one HPC from IBM made in 2001 with 12 teraflops of computational power and distributed to 500 nodes, and maybe many of you have an iPhone in your pockets, released in 2022, and it has already 1/6 of this computational power. So all of you are literally sitting on an HPC computer. It should result better gene trees. You could, for example, calculate nuclear explosions on your phone or use a model, yet you probably play Plants and Zombies. But the problem is not just you, so not just human error. Let's check the model, how we are inferring gene trees. Usually you have some type of alignment, and you have a substitutional matrix, a substitutional model, and you are throwing gene tree proposals, like topology proposals, and you calculate for all of these a probability, a likelihood. And the good thing is that you don't have to do it by hand. There are plenty of programs to do it, even RefBase. But does this pipeline model biochemical constraints properly? You could ask. Let's take the calcitonin receptor, which is a transmembrane protein. I'm not expert in it, but I just found it a nice example. And if you try to match the inter-membrane and parts of the alignment, and you color the amino acids by their biochemical property, then you would assume that maybe it's important that what was hydrophilic stays hydrophilic, what was hydrophobic stays hydrophobic. And you would expect from your evolutionary model that it will model it appropriately. You would expect this actually from the substitution matrix. This is not this is a Blossom matrix. This is basically a penalty matrix, but from this you will have the substitution matrix. And what we saw before that, for example, this K at site 249 is in the interior, and at 280 there is a D amino acid. So here you see the penalties of changing this amino acid to another amino acid, and you are actually happy because you see that, okay, this K can change into R and E and Q, whereas the D at 280, site 280, can change to N and some other sites. And Lénard Szantho, one of our co-authors, did a nice blog post, which I recommend you to read, actually a couple of years ago already, where he tested that whether these constraints are kept after multiple substitutions. And on the first line, everything is fine. You have D and K. After one substitution, you are still happy because it's changed into something with similar biochemical properties. But after 3 or 4 substitutions, so on a longer branch length, then actually the memory is lost. So your simple model didn't keep this biological constraint, which may have been important for this protein to function properly. And this per site amino acid frequencies we can summarize with a effective amino acid frequency per site number, which for which you see the equation above. So again, you have the normal pipeline with a site homogeneous model, but you have the knowledge from Lénard Szantho and from me that your amino acid frequency is actually the global amino acid frequency is not necessarily the one which your site is constrained to. So it's not appropriate if you have long branch lengths, so deeper phylogenies, to model them with simple models, but you need a site heterogeneous model. Otherwise, you will run into systematic biases, for example, long branch attraction. You can make a simulation. On the left side, there is the tree along which we simulated. We simulate under a site heterogeneous model, then infer with a site homogeneous model, and we get a different tree, much possibly. So the take-home message is that you should use site heterogeneous models, see XX, CAT, ED cluster, and my talk could be finished here. But you would then ask the question that, okay, what is then CXS is not working, and what is when your phylobase is running since 3 months and still didn't converge? This is when you should use our method, the CAT PMSF, which is both a way to detect LBA and also a much faster method than just using phylobase alone. How does it work? You have your alignment, then you make with any software, just make a tree. It doesn't have to be the best tree, but just make a tree, for example, with a site homogeneous model. Then you give this tree to Phylobase or some other software which can then calculate the site-wise amino acid frequencies, like a site profile for the amino acids, and let it run. It will take some weeks, but not too much. Then you export these site-specific profiles, and at the end, get another tool. We use IQ-tree in our paper and now we let the topology free, but we give to the model, the exchangeability model, we give this from Phylobase inferred site profile data, which will make it better than C60, for example, because we have much more categories to mix. We first did simulations just to check the whole process. So the branch lengths we iteratively made a bigger difference between them, up to 2, and then we inferred with site homogeneous, site heterogeneous models, and we calculated the site-wise log likelihoods, because this will be necessary for the so-called compositional constraint analysis, where you take 2 topologies. You see that the 2 trees are different there with the same model, and export this site-wise likelihood. You take the difference, and you also have from Phylobase or other tool the site-wise effective number of amino acids. and then both are sidewise, so you can make a effective amino acid versus log likelihood difference plot, and this is what we call basically compositional constraint analysis. You can bin it by effective number of amino acids, then you get this plot, where this will show you the support for one or the other topology. So the upper, the blue part is then the one of the topologies, and the yellow part is then the other topology. And you can measure the support for one or the other. The other version of this plot is the cumulative, and you take the cumulative difference. The good thing is that then you have the final likelihood, what actually the IQ tree would give you. But it masks the per key effective bars a little bit. So, having all these tools, then we can investigate. Let's do this simulation. First, we have almost the same branch lengths. P and Q are very close. And you see on the bottom plot the site heterogeneous models, and on the lower plot the site homogeneous, plus a C60 model. And what you see that both are fine. They support consistently the same topology, the blue one. If you just have an 8-fold difference between these branch lengths, then the site homogeneous models start to give you inconsistent signal. So the low key effective sites are supporting one topology, whereas the higher number of key effective sites support the other. And if you increase the difference even more to 12-fold difference, then the site homogeneous models completely fails to detect the recover the initial topology, and all site heterogeneous models do. You may ask for the red bars on the top right one. Those were the LG exchangeabilities, and we simulated with Poisson, so that's why the LG could never learn Poisson properly, but GTR and Poisson could. So let's anyway go to real-world examples. There are some well-established LBA examples or papers. For example, from Philippe et al., the case for nematodes and platyhelminths. On the left side, you see the LBA prone tree, whereas on the right side, what you would recover with site homogeneous models. Whereas on the right side, you see the hopefully LBA free, so the common consensus trees as of today, which you would recover with site heterogeneous or more advanced models. And indeed, if you check the compositional constraint plots, then you see, I actually prefer the cumulative ones. So you see that the LGC10 model still recovers actually the wrong topology, and only above C60 you would get the good one with for platyhelminths. Whereas for nematode, actually even using a proper model of with C10 is enough to recover the proper topology. Whereas just using LG or GTR is not enough. You will get inconsistent signal, and it will tell you very surely that the wrong topology is the good one. Another example was the microsporidia. You see again here the question was for a long time whether microsporidia should be at the sister for all other eukaryotes or be at the fungi, sister to fungi. And Brinkman et al. Had a paper about this in 2005. And again, you see the same pattern, but actually here the site homogeneous models perform much better, except the LG and GTR, but here again, all the C10, C60 models could recover the proper topology, and also, of course, our CAT PMSF model. Okay, a more controversial example is the placement of Ctenophoras and Poriferas. Actually, I am now, I think, in the territory of the Ctenophora believers, and for where I come from, Europe, we are more for Porifera. And it's an interesting result, I think, for this debate. We used the Simeon et al.'s dataset, which was a huge one. And if you observe, then until now the log likelihoods were like pretty high. No, it's only 10 maximum, and 25 with the cumulative one. So it's not a big difference, but still the pattern is similar. That whereas the site homogeneous models, they prefer one of the topologies, and all models are preferring the other, the poriferous sister one. But it's so the site homogeneous models are just, I mean, with this 3000 likelihood, they are like very, very sure in the other model. So it still requires further investigations to arrive to a clear conclusion. But our take, based on our examples previously and or the behavior of the models, suggests that poriferous sister may have been the proper one. So, because we have some time, whenever you would like to actually use our model, then I show you some examples of inconsistent signal. So whenever you would see your model support one or the other, for the low key effective sites, a different model than the high key effective one, then you should suspect for LBA. This was Simeon et al., so the previous dataset, but if you include all the outgroups, which usually, if you include distant outgroups, then it will increase your LBA effect. So this was expected, and there is also a Ryan et al. Dataset. In the supplementary you can find it, which is also investigating tinophorand porifera placement. And here you see that the log likelihood difference is just 1.5, so it's just too small to give a conclusion. But it's the same for the C60 model. So with that, I thank you for your attention. And
thanks for coming. So I'm Megan Smith, and I'm an assistant professor at Mississippi State. And today I'm going to tell you about some work that we've been doing about using paralogs in phylogenetics. And this is work that I've been doing in collaboration with Mahan. So typically in phylogenomics, our pipeline is to start with a set of coding sequences for the taxa that we're interested in, and then to use some clustering program like OrthoMCL to get ortholog groups. These ortholog groups are groups of closely related genes and include both orthologs, or genes related through speciation events, and paralogs, or genes related through duplication events. So for example, here in the first tree there, we have an ortholog group where there's a single gene from each species. And here in the second tree, we have an ortholog group where there are 2 copies in species A and species C. And the red stars here are indicating that these copies are related through a duplication event. So this ortho group includes paralogs. Most commonly in phylogenetics, we try to use orthologous sequences. And so often we rely on these single copy genes, where there's just one gene sampled from each species, because it's thought that relationships in orthologs will reflect speciation events. However, this involves throwing out a really large proportion of our data. So more recently, there's been a lot of talks about using both orthologs and paralogs in phylogenetic inference. And so we've published a few things recently about doing this. So we've used some simulation studies as well as analyses of empirical data sets to demonstrate that you can accurately infer species trees while using these larger gene families. And this is particularly true with methods like Astral that work well with discordance. It's not just us saying this, so there have been other people looking at this as well, and it seems like we can accurately infer species trees with these larger gene families. So most of this previous work has really highlighted the ability to use more data as the primary reason to use these larger gene families for inference. But today I'm going to talk about another potential benefit of using larger gene families. And to do that, I'm going to talk about something that you just heard about in the last talk as well, long branch attraction. So for long branch attraction, we have, in our true tree shown here on the left, we have 2 taxa, B and D, that have long branches. And because of homoplazy, this will often cause us to incorrectly infer a tree where B and D are sister to each other, even though that doesn't reflect the relationships among species. a really common solution that's been suggested for long branch attraction is to add taxa. So if we can sample a taxon like E that breaks up the long branch leading to B, and we could sample a taxon like F to break up the long branch leading to D, that can help mitigate the impacts of long branch attraction. Of course, that's not always easy to do, right? There might not be a taxon that we can sample. We might not have money to get additional sequences from these taxon. There can be various reasons that this might not be a viable solution. And so what we're proposing is to, instead of adding taxa to break up long branches, we could add genes to break up long branches. So here, the red stars are indicating duplication events. So we can sample a gene where there's been a duplication event in species B, and this can break up the long branch leading to B, and there could also be duplication events in D, which would break up the long branch leading to D. And this is great because we already have these genes, we just usually throw them away, so let's try not throwing them away. So to evaluate whether this would help with long branch attraction, we did some simulations in SimPhi, and we used a substitution rate modifier Q to ensure longer branches in B and D. Importantly, this is only affecting the number of substitutions occurring on these branches. There are not more duplication and loss events occurring on these branches. And so we simulated these data with duplication and loss. We inferred trees with maximum parsimony and maximum likelihood, and then we estimated quartet concordance using Astral. So we estimated the proportion of quartets that are supporting this relationship, the true relationship where A and B are sister. Okay, so these are the results for just our single copy genes. So this is the standard result, and this is with maximum parsimony. Q here is the multiplier, so the longer Q, the more long branch attraction we should have. And R on the x-axis is the length of the internal branch. Dark colors indicate that we did not find high quartet support for the species tree, and light colors indicate high support for the species tree topology. As we expect, as Q gets longer, we get lower support for the species tree topology, and as R gets shorter, we have lower support for the species tree topology. So this is just single copy genes. Let's add all of our gene families in. And so here, what you can see is that we have a lot more yellow and purple and a lot less black here. So we have higher support for the species tree across a lot of the parameter space here. And that's just, so more of our quartets are supporting the species tree topology. So using these gene families with paralogs is helping to mitigate the impacts of long branch attraction and maximum parsimony. We see a similar pattern in maximum likelihood, although it's less pronounced because likelihood is doing better than parsimony and with long branch attraction, as we expect. Okay, so that was a set of relatively simple simulations. We're going to make things a little harder for ourselves by adding this outgroup here. And when we add an outgroup, we have opportunities for duplications in the ancestor of our focal taxa. So we can have a duplication on this branch leading to A, B, C, and D. And this can lead to additional discordance. So this can lead to gene trees like this, where we have two copies. Each copy is present in all four of our species. And some of the quartets that we can sample from this gene tree will match the species tree, like this. So we could sample one copy from all four species. That's great, matches the species tree topology. But we may also sample quartets that don't match the species tree topology. So if we sample this quartet, which spans the duplication node, we're going to end up with A and D sister to each other and B and C sister to each other. So we're adding noise, and we add more noise the longer that branch is. Okay, so here on the left, I have the results from maximum parsimony on single copy genes again. And as Q gets longer, we do worse. There's no relationship here with the ratio of the outgroup height, which is what I'm showing on the x-axis. But when we use all of our gene families, we can see that the higher that ratio is, so the longer that internal branch is where we can have those duplication events, the more noise we're adding. So we are doing worse as the outgroup gets longer, and we have lower support for the species tree topology. It's not necessarily all or nothing, though, so we don't have to just use single copy genes or use all of our gene families. Instead, we can focus on a subset of gene families. So here in this middle tree, we have duplication events, but we only have duplication events in the ancestor of B and in the ancestor of D. So these duplication events break up our long branches without adding any additional noise. And so we can focus on these genes for inference. these gene families with only lineage-specific duplicates could further improve inference compared to using all gene families. And that's exactly what we see. So these are results from maximum parsimony again. So on the left, we have results from single copy genes, and on the right, we have results from these lineage-specific duplicates. And we can see that we have high quartet support in a larger region of parameter space. So we're doing quite a bit better. So using these lineage-specific duplicates mitigates long branch attraction without adding this additional noise. Okay, so that's great. This works on simulated data. What about empirical data? To evaluate this on empirical data, we use data from pseudoscorpions and scorpions. So long branch attraction has been blamed often for the failure to recover scorpions and pseudoscorpions as sister taxa. So in many studies, pseudoscorpions are recovered as sister to scorpions, but when taxon sampling is worse, pseudoscorpions are recovered as sister to other long branch taxa in Acariformis and Parasitiformis. And so because of that, this has been attributed to long branch attraction. So we used whole genomes from 18 chelicerates, including scorpions and pseudoscorpions, and we used fast ortho for ortholog clustering. We then constructed gene trees with parsimony and likelihood, and we built 3 different data sets, one with our single copy genes, one with all our gene families, and one with gene families that have a duplication in pseudoscorpions. We then calculated quartet support for scorpions and pseudoscorpions as sister taxa using Astral. Okay, so with our single copy genes, about 14.6% of our quartets support this relationship, so very low with maximum parsimony. We see that a higher percentage of quartets support that relationship in likelihood, as we would expect if this is caused by long branch attraction. When we use all our gene families, we see a slight increase. So we go from 14.6% to 15.7% in parsimony, and from 18.7% to 19.8% in likelihood. And when we use lineage-specific duplicates, we don't see any further increase in parsimony, but in likelihood, we see a slight increase from 19.8% to 20.5%. However, these numbers in parentheses are confidence intervals, which are overlapping. These end up being some fairly small data sets in some cases. So support for scorpions and pseudoscorpions does increase as we add data from more gene families, but we have these overlapping confidence intervals. Okay, so overall, long branch attraction misleads inference in phylogenetics. That's not new. Using data from larger gene families can help mitigate this because paralogs can break up those long branches. And overall, using data from larger families for phylogenetic inference lets us use more data without sacrificing the quality of our inference, and can even improve inference in some regions of parameter space. Okay, so thanks to you for coming. Thanks to my lab, which is recruiting students for fall 2025, and I should have time for questions.
Hi everyone, I'm Analisa. I'm a third-year PhD student at the University of Connecticut, and today I'm going to talk about how we've been testing our phylogenetic method, the sequential multi-species coalescent. I'll start with some background on the model that we're using. The multi-species coalescent provides a Bayesian way to estimate a species tree in the face of deep coalescence. Here we have a species tree where A and B are sister and C is more distantly related. This pink gene tree represents one example of a gene history we could have within this species history. Here, the topology of the gene tree matches the topology of the species tree. On the other hand, we could have something like this purple gene tree. Now the topology does not match the topology of the species tree. If we were to make a tree from just this one gene, we would be misled as to the true history of these species. One reason this can happen is called deep coalescence. Deep coalescence occurs when gene lineages coalesce below the bounds of a species tree. Below that dashed line, the gene lineages are unconstrained and can do whatever they want, which can cause these topological conflicts. The multi-species coalescent aims to take into account all of these gene histories to estimate one species history. I'm going to talk about a couple of parameters later in my talk. The first one is T, or species tree height. That's the height from the tips of the species tree down to the most ancestral speciation event. We also have theta, which is the distance between two taxa in terms of expected number of substitutions. Now I'll give a really brief overview of how our method works. Our method uses something called sequential Monte Carlo, or SMC, to sample from the multi-species coalescent posterior. In our method, we have a bunch of particles, and every particle contains a species tree as well as all of its associated gene trees. On this slide, the box represents one particle, but you can imagine scaling this up to many particles. In this example, we have one gene tree, which is represented by those 5 dots. Those are the 5 taxa at the tips of our tree. You can see that at this first stage, that tree is completely unresolved. Those taxa are totally disconnected. We also have a species tree with 2 species, and again, that tree is also unresolved, so those 2 species are also disconnected. Every species population is associated with a theta, since that's a parameter we're interested in estimating. We then begin building up our trees. We start out by drawing an increment and adding it to all of the lineages in existence in all of the trees, so the species tree as well as all of the gene trees. We then choose either a speciation event or a coalescent event, and we choose either 2 species or 2 gene lineages to join at random. I'm now representing the species tree as just that thick black line. That line represents a barrier to gene flow, so under this model, gene lineages cannot cross that barrier. Next, we give each particle a weight, and we filter the particles according to those weights. A particle's weight is equal to the likelihood after the join divided by the likelihood before the join. Particles with heavier weights get resampled and copied to the next generation, while particles with lighter weights get filtered out and we don't see them again. This is just like natural selection, where particles with lighter weights essentially have lower fitness and can't compete with the other particles. We then continue these steps, so I'm showing here that we're again drawing an increment, joining either species or gene lineages, and then we filter at each of these steps. And now I'm just showing that we're continuing those steps until we have a completely resolved species tree as well as fully resolved gene trees. At the end of all of this, every particle represents a sample from the multi-species coalescent posterior. At this point, we noticed that we were having some problems with particle degeneracy. Particle degeneracy is when one particle carries all or most of the weight at a certain stage, and that particle essentially dominates, and we get these bottlenecks where we lose a lot of variation in our particles. At the end of the first round, even though we have a good species tree, often we have very little variation in that species tree, and since this is a Bayesian method, we're interested in getting a distribution of species trees. The way that we've worked around this is to take the gene trees that we estimated from the first round, wipe away the species trees, and rebuild them conditional on those gene trees. I'm showing here 3 gene trees that we estimated from the first round, and now I'm showing how we're building up species trees conditional on those gene trees. We filter the species trees now by the coalescent likelihood, which is the probability of a gene tree given a species tree. You'll notice in that second particle, I've proposed a species barrier that's illegal under this model. It crosses the gene lineages, which can't happen according to this model. That species tree gets a coalescent likelihood of 0, and it does not get chosen in the resampling. So instead, here we choose to resample particles 1 and 3. The coalescent likelihood is less decisive than the Felsenstein likelihood that we used in the first round, and so this alleviates those issues with particle degeneracy, and at the end of this second round, we have much more variation in our species trees. Now I'll talk about how we've been testing our method. We wanted to compare the performance of our program to the performance of StarBeast 3, which is an MCMC-based multi-species coalescent program. We wanted to do a pretty thorough simulation study, and so we conducted a bunch of simulations, each with 10 loci, 5 species, and 10 taxa. These cubes are going to display the results from those simulations, and so I'll set up how that's working. The x and y axes of these cubes will show the parameter values that we use for each simulation. We chose 10 evenly spaced values between 0 and 1 for t, or species tree height, as well as theta. Then we took every combination of these parameter values and we conducted a simulation at that combination of values. At the end of this, we had a grid of simulations that covered a wide range of parameter space. The z-axes of these cubes will show the Robinson fold, or RF distance, from each of the programs to the true tree. In the first cube, that axis will show the SMC RF distance to the true tree minus the StarBeast 3 RF distance to the true tree. An RF distance, or Robinson folds distance, measures how similar 2 trees are. A distance of 0 means that the trees are identical in terms of topology. In the other cube, that z-axis is just flipped, so we'll have the Starbase 3 RF distance minus the SMC RF distance. Both of these cubes will show results from the exact same set of simulations, but by flipping that axis, we'll be able to see both sides of the plane of simulations. Here's how we chose the settings for both programs. For our program, we used 10,000 particles for the first round. For the second round, we conditioned on 5% of those gene trees, and then used 200 species particles per set of those gene trees. For Starbase 3, we used 6 million iterations with 600,000 iterations of burn-in. I wanted to make one comment here about time. So one thing that's attractive about SMC-based algorithms is that they're easy to parallelize. In our program, we take the particles and we spread them out across all of the available computer processors. This lets us build up particles in parallel, which can significantly speed up runtime. However, it's really hard to do a fair speed comparison between the two programs because it's easy to just run one program a little longer than the other and make it seem like the other program is really doing much better in terms of speed. Because of this, we chose settings so that both programs took about the same amount of time without any parallelization. We felt like this was the fairest way to give both programs kind of an equal shot at getting the simulations right. But because of this, we don't really have results for speed, even though our program can be sped up significantly with parallelization, especially if we have access to a lot of computer processors. Okay, so finally, here are the results from the simulations. Again, in that first cube, the z-axis is showing the SMC RF distance to the true tree minus the Starbase 3 RF distance. This means that points above the plane are simulations where our program performed worse than Starbase 3. In the other cube, it's the exact same results, except that axis is just flipped, so we're seeing the underside of the plane. So points above the plane are simulations where Starbase 3 performed worse. When we did an ANOVA on these results, we found there was no significant difference between the performance of the two programs. When we just look at the planes of simulations, we also don't see one region of parameter space where one program is consistently outperforming the other. Instead, we see some simulations where Starbase 3 performed better, and some simulations where our program performed better, but it doesn't look like there's any systematic bias towards one part of the plane, and so that's another good sign that the performance of the programs is comparable. I'll end by talking about a couple of empirical examples. For this first data set, we chose a data set with 9 gopher species, 6 genes, and 27 taxa. I'm showing here the consensus trees from a Starbase 3 run as well as an SMC run. I'm also showing the bootstrapped SVD quartets tree. SVD quartets is not a Bayesian program, but it's very fast, and so it provides a really good comparison for the other programs. The outgroup for this data set was supposed to be heterodis, but you can see that neither Starbase 3 nor SMC recovered that, and SVD quartets only shows it as the outgroup because we had to route the tree that way. When we look at all 3 trees, we mostly see that the programs are recovering the same 2 major clades with some differences. If you look at the SMC tree, you'll see that one species, bulbivorous, is down at the bottom, and that's because we could not resolve the placement of that species. It jumped around between the 2 clades so much that it didn't actually show up in either clade in the consensus tree, so that was one key difference. I'm now showing the consensus tree of those consensus trees so that we can get a better sense for which programs are supporting which groups. Again, like I just talked about, all of the programs disagreed on the placement of the outgroup, heterodis, so that's down at the bottom. All the programs did agree on that bottom clade, so they all got those 4 species in that second clade together. We do see some disagreements in that top clade, so it seems like that top clade was a bit harder to resolve. Part of that is due to the disagreement in the placement of heterodis, like I talked about, and also because our program could not resolve the placement of bulbivorous. And so overall, it seems like this may have been a little bit of a difficult data set to resolve. Lastly, we tried a slightly larger data set. So this data set contained 7 rattlesnake species with 15 genes and 52 taxa. Now when we look at the trees, we see that they all recovered the outgroup, and they all recovered the same 2 major clades with very high support values, so that's a good sign. Here is the consensus tree of those consensus trees, and so this time we see that all the programs got the same consensus tree with hundreds for all of the branch support values. And so it looks like maybe that first data set was a little bit trickier to resolve than this one. In conclusion, I've just talked about how we've been testing our phylogenetic method, the sequential multi-species coalescent. This is a fully Bayesian but potentially efficient way to sample species trees. And so far, based on our testing, it performs comparably to StarBeast3 on both simulated and empirical data. I'll end by thanking the NSF for funding, as well as the STORES HPC cluster for allowing us to run the analyses, and I think I have time for a couple of questions.